here we go again with video number 4. So, we just talked about reproduction, and now we're going to move on to talk about the planktonic larval phase. So during this time, the larvae are often carried out to sea. They're changing substages and metamorphosing their shape, that's what that means, and then immigrating, if they're lucky, back to the reef, back to the suitable habitat. And while all of this is going on, they have to deal with oceanographic transport. So oceanographic features like currents, like gyres, like fronts, all kinds of things happening and moving them around. Larvae can move a little bit, but not as fast as most of the oceanographic features. They also have to deal with predation, being eaten by other organisms. They have to deal with starvation if there's not enough food. They have to deal with physiological stress, if the temperature's wrong, if humidity is wrong, or whatever. So let's go ahead and talk about it. Okay, so what do these larvae things look like anyway? Okay, so this is the non-feeding larva of a sea star. Okay. So this is the non-feeding larvae of the sea star. It's a really cool looking sea star. It's one that is found here in this area, and you can see how big it is next to that key. This is a larvae of a bryozoan. Again, one that's found in this area. The non-feeding larvae. Sorry, I'm getting confused here. This is the veliger larvae of a gastropod snail, and it's unidentified, so I just put that on there. This is the unhatched tadpole larvae of a sea squirt or tunicate, which is in the chordata like us, and in the class Acidia. So sometimes it's called Acidiaceae. I don't know how you say that word. Anyway, and here's the feeding larvae of the sea urchin, Strongylocyntis perforatus, the purple sea urchin that we see fairly often. And finally, the feeding Brachylaria larvae of the sea star Pycnoglodia helianthrides, which is the sunflower star. So some of these, like this one, is a non-feeding larva. So if it can't feed when it's in the water column, it's not going to be there very long. However, I think the rest of them are pretty much all feeding ones, so they can all be in the larvae in the water column for a longer period. Okay, so here's what it looks like for corals specifically. If they're brooders, then they'll release the planulae, which is the larval form for corals, into the water column. If they're external fertilization, then they'll release the eggs and sperm, hopefully synchronously at the same time, have fertilization, which forms a zygote, and then again the planula gets into the water column. And then eventually the planula, in a couple days usually, is ready to settle, find the appropriate habitat, and it settles and forms one primary polyp. And then you get that body, and then I talked about earlier, that forms a colony growth. So for this coral, which is again the UC Santa Cruz, this is what an early colony looks like. This is maybe 20 or 30 polyps. This is after it's died, it's been dried. But you can see it's a very small colony at this point. And then there's an even smaller one over here. It's maybe only like 3 or 4. Okay. So, what are the two main types of larvae? Like I mentioned there, some larvae can feed, and therefore they can stay in the plankton a lot longer. Some larvae don't feed, so they can't stay in the plankton very long. So planktotrophic larvae, imagine that, can eat plankton. And they are feeding larvae. And so the organisms that do this, they produce many, many, many eggs, hundreds of millions of eggs that are really small. And they don't have much yolk, so they don't take much energy to make them, so that's why they can make so many. And they tend to be in the plankton for a really long time, like weeks or months. And so they're doing most of their developing when they're in the plankton. Whereas on the other hand, the pseudotropic larvae don't feed. They have to survive on just what's in that yolk. And because that yolk is a big energy contribution from the parent, they can't make as many eggs, so they make maybe tens, hundreds, thousands of eggs, instead of that hundreds of millions. And their eggs are larger, again with more yolk. And because they're dependent on that yolk and they can't be in the larvae very often, in the water column very long. By the way, planktonic larval duration is a term we'll be using a lot. and we often shorten it to say PLD. Acetotrophic larvae, actually, a lot of the development can happen when they're being brooded, when they're, still being held by the parent in some way. Sometimes in a capsule that's laid on the bottom of the ocean, in the benthos. And sometimes it's pentopic, depending on the species. Okay, so planktotrophic larvae, because they have that long PLD, can disperse very far because they have to feed and they don't have that yolk that is providing like a backup. They tend to have very high mortality, but also because they don't have that yolk, it doesn't take much energy for the parent to make that egg. So one example of that is our butterfly fish. They make planktotrophic larvae. So I looked it up. This particular species, their larvae takes about 100 days before they settle out. So they definitely have a pretty long planktonic larval duration. I mean, that's more than 3 months. So with acetotrophic larvae, because they have a short PLD, they have a relatively short dispersal range. Because they have that yolk and they have a short dispersal range, they have pretty low mortality. A lot of them are going to survive. But because they have that yolk, it takes a lot of energy for the parent to make each egg. So one example of this is our feather duster worm, which produces a larvae that looks like this. Well, this is one of several stages. And their eggs are between 150 and 200 microns, so greater than 100 microns. And their PLD is only about 6 to 8 days, much, much, much less than butterfly fish. So that makes sense that they have that short PLD that's characteristic of the acetotrophic larvae. Okay, so while these larvae are in the water column, they have to deal with getting carried out to sea, sometimes, getting back in if they do, going through all these different stages where they're changing constantly. They're getting carried around, they're getting eaten, they may not have food, they might be starving, they're getting stressed out by what's going on. They're dealing with so many different things that it is not at all surprising that there's pretty high mortality. But while these larvae are getting carried around, they either can be staying pretty close to their population, or at least get carried back to their population, so that there's little or no exchange between different populations, or the populations can be really pretty open and get carried as much between populations as they do within a population. Of course, these are the two extremes. There's also in between, where they're staying somewhat in the same area and some exchange with other populations, right? So it's really important to understand that exchange between populations when we're trying to establish marine protected area networks. Remember, Angela talked about whether you want to have a single large reserve, several small reserves, or reserves that are connected. So you really have to know what's going on with the larvae and connectivity in order to plan this out and do a good job of it. And also you need to understand what's going on with the larvae so that you'll know whether the population you're protecting is a source population that's producing lots of young, or is the same population where everything's dying and it's not contributing to the other populations. Okay, so most larvae and also the dispersive stages of algae can swim, but pretty slowly, usually less than 1 millimeter per second. So in most cases that means that the oceanographic features are actually moving the larvae faster than the larvae themselves can swim. So for most of the history of marine ecology, people have modeled larvae as just passive particles. You can use a drifter, you can use some dye to just figure out where the larvae would be going. Now we know that some fish or some larvae actually do have real behavior, that they're responding to cues. They can go up or down to find the water body that's moving the way they want to move, in order to, go where they need to go, whether that's out to sea or back to shore on their way to settle. And some larvae, particularly fish larvae, can actually swim quite a bit faster than 1 millimeter per second. And so they really can control where they're going to some extent. They're not just passive particles. Okay, and finally, obviously, most larvae are way too small for us to see with the naked eye, and they're in the water column too long for us to observe them directly. It's really hard to study larval dispersal in the field and what's going on with larvae in the field. So we'll be talking a little more about how on earth we can do this. And this has just been happening in the last 10, 15 years that we've been able to figure out ways to study larvae in the field. Before that, it was all in the lab, or we just assumed it wasn't important because we couldn't study it. Okay, so from the lab work, we do have some idea of the pelagic larval duration of a lot of fishes, and I believe that's probably how they estimated this dispersal distance for fishes, invertebrates, and seaweeds. So if you look at it, seaweeds, their dispersal distance is usually between about 10 meters and about 1 kilometer. So they're not dispersing that far. Invertebrates, on the other hand, are going from about maybe 50 meters up to maybe a couple hundred kilometers. So invertebrates are going pretty far. Fishes are going from maybe 5 kilometers up to maybe even 1,000 kilometers. So their dispersal distances can be pretty far because they have relatively long pelagic larval durations. However, we're getting a better idea of this than I think this paper presents. Okay, so organisms produce hundreds of millions, or at least tens, of larvae, like every year, maybe multiple times a year. But there are a hell of a lot more larvae in the ocean than there are adults. So even if we can't study them very well, we know larval mortality has to be humongous. It just has to be, because otherwise there'd be a lot more adults. So what are the factors that really increase larval mortality? There's physiological stress, wrong temperature, wrong salinity, too much light, all kinds of things that I'm sure you can think of. Starvation, sometimes there's not enough food for them, and especially the ones that aren't given the yolk, that have to make sure they find enough food on their own. That can be tough. Predation. Apparently benthic invertebrates don't actually apparently have that much impact on larval mortality, but fish and pelagic invertebrates, like the Cnidarian medusa and jellyfish and ctenophores, they do eat a lot of pelagic larvae. And when they're coming back to the coral reef to recruit, people started to have called the reef the wall of mouths, because if you look at it, there's corals that are filter feeders, there's all these planktivorous fishes up in the water column. There's so many organisms there that are trying to eat the plankton that are coming in to recruit. There really is a wall of mouths I have to get through in order to settle and find them in the spot. The larvae also have to deal with sinking, because usually, you know, they're pretty buoyant, but, you know, not 100%. So they're still going to sink eventually, and they can be carried by oceanographic features like currents away from their appropriate habitat. So that can also be a problem. If they can't get back to their appropriate habitat, they're in trouble. But larvae do have ways to combat a lot of that. So first of all, a lot of organisms have evolved to release their larvae at times that can minimize that physiological stress, minimize that starvation, minimize that oceanographic transport away from the appropriate habitat. They can deal with predation by swamping the predators, just releasing so much larvae and eggs and sperm that the predators just can't deal with it. So remember that mass spawning of corals, that's really what was going on. Some larvae also have morphological or chemical defenses against predators, and larvae can also respond to cues like light or temperature or tide to make sure that they're not getting carried away from their appropriate habitat, maybe sometimes even to avoid predators, find appropriate food. So why would larvae evolve to have such a low chance of survival and such a low chance of finding suitable substrate where they can live? Seems like a humongous waste, right? What would the benefit be? Well, it's all about dispersal, really. So, people hypothesized that the main benefit of that dispersal was that at least some offspring would be able to find and occupy suitable habitat, even if that parent's patch, where their parents lived, becomes unsuitable because of some sort of disturbance, or there's, you know, too many competitors already there, or there's predators there, or something. So it's basically the idea of spreading the risk. If you produce enough babies, at least some of them will survive. So how would you test this hypothesis? Kind of a tough one. So this guy, Jablonski, in 1986, did this really cool study where he looked at the fossils of Cretaceous gastropods found at these different outcrops